Hello everybody, my name is María José Díez and I'm going to talk about plant traits in this first presentation of the training course on tomato phenotyping. The tomato plant consists of one stem, which is long and thin. Leaves develop along the stem as well as the inflorescences. The flowers are of a beautiful bright yellow. The tomato fruit itself can be variable in shape, size, and color. The typical mature fruit is red and round, but it can also be orange, pink, yellow, or even purple. The size of the tomato also varies. Cherry tomato plants produce very small tomatoes, while other varieties can produce tomatoes much bigger. Finally, the root lives underground and provides support and nutrition to the plant. The architecture of the tomato plant is the result of the expressions of many genes and the effect of the environment. The different parts of the plant can vary greatly depending on the environment in which the plant is growing, open air or greenhouse, the temperatures, the solar irradiation, and the nutrition and watering. In this video, we will learn how to phenotype the main characteristics of the plant. Basically, we will focus on the presence of anthocyanins on the plant, the growth type, the length of internode, and the plant age. Not all these traits will be evaluated in the phenotyping trial. This will be indicated in each slide. Anthocyanins are the main water-soluble pigments widespread in many flowering plants. The red, blue, or purple colors found in various plant tissues, including flowers, leaves, fruits, and seeds, are mostly attributed to the accumulation of anthocyanins. Anthocyanins play essential roles in protecting plant tissues from being damaged by light, ultraviolet radiation, cold, drought, nutrient deficiency, and pathogens attack. Anthocyanins also act as important health-promoting supplements in the human daily diet. In tomato, anthocyanins can be present in different parts of the plant: leaves, stem, and fruits. Black tomatoes owe their color to the accumulation of anthocyanins in the peel. The first trait we are going to take is the anthocyanin coloration of hypocotyl. This trait is recorded only in seed-propagated varieties. It can be absent or present, as you can see in these pictures. This trait is controlled by the gene AA, anthocyanin absent, and the phenotype is the absence of anthocyanin in all parts of the plant. As we have seen, the anthocyanin can be present in all parts of the plant. For this reason, it is also taken in the stem. In this case, we can have different intensities, from absent or very weak to very strong. Most of the varieties are classified from 1 to 5. Expression of anthocyanin is influenced by day temperatures. Under greenhouse conditions, the variation is rather low. You can see some examples in this slide. You can also see that anthocyanin coloration can be not homogeneously distributed, being very dependent on some environmental factors, especially of light intensity. Now we will focus on the architecture of the plant. Aerial plant architecture is predominantly determined by growth habit, shoot branching, and leaf morphology. All are genetically controlled, and the combination of these genes can produce plants that look very different. The genes SP, blind, and lateral suppressor control the plant architecture, while other genes control many aspects of leaf development and complexity. We will focus in this presentation on the architecture of the plant. One of the main traits is the plant growth habit. Plant architecture can influence yield by altering the relative growth and position of vegetative and reproductive organs in the plant. In most Solanaceae, there is an alternation between vegetative and reproductive phases, which present themselves as modular units called sympadia. The vegetative apical meristem converts into flora after a series of 5 to 12 internodes with leaves. Vegetative growth, however, continues through the top meristem, which grows, displacing the inflorescence to the side and producing a new sympodium with 3 leaves and an inflorescence. This process is repeated indefinitely in plants of indeterminate growth habit. Determinate growth in tomato is a consequence of a recessive mutation in the self-pruning Sp gene. The loss of function Sp allele reduces the number of leaves of successible sympadia units, resulting in 2 consecutive inflorescences that top vertical growth, producing a compact plant. Semi-determinate varieties do not have consistently 3 leaves or internodes between inflorescences, and show semi-determinate growth, for example, with the termination of the stem with the 9th inflorescence or at higher than 20 inflorescences. Here you can see the typical structure in the point of determination of a tomato plant of determinate growth habit. In the case of semi-determinate growth, it exists an initial point of determination, but the vegetative growth continues producing more leaves and inflorescences. In the indeterminate plants, continuous development of leaves and inflorescences occurs. Determinate cultivars have synchronized flower and fruit production, which allows mechanical harvesting in the tomato processing industry, whereas indeterminate ones have more vegetative growth with continuous flower and fruit formation, being thus preferred for fresh market production in the open air or in the greenhouses. For a given plant, growth type, there exists variation for the distance between inflorescences. This trait is named length of internode and should be observed or measured at what time for the whole trial. For example, after a fruit set on approximately 5 nodes. The total length of the stem should be observed or measured between the first and the 4th trusses. In case of measurements, this measure is divided by the number of internodes in between, and an indication of the length of the internode is given. In this slide, you can see the high variation that exists for this trait between plants. Other trait measured in plants with growth type indeterminate is the plant age. It should be measured at one time for the whole trial, for example, 60 days after planting, or after a fruit set on approximately the 5th inflorescences, or when the first variety in the trial has reached the wire in the greenhouse or the top of the stake. There is also a high variation of this trait between plants. Thank you for your attention. I hope this video has been interesting for you.